Good evening, sir.
Good evening.
Sir, you can start.
Yes, ma'am. Okay, thank you, ma'am. Good evening, my dear students. Good evening, Dhanalakshmi.
Good evening, sir.
Good evening, Pa. Okay. So, once again, you all are welcome to the biological classes. Okay. So, last class, what we discussed, there are two types of vectors. One is natural, another one is called as artificial. So, here, we are going to discuss about in deeply what is meant by artificial vectors, how they constructed, who is discovered, and where it discovered. All those things already we discussed in the previous classes. Okay. See that the examples of artificial vectors are the plasmids. The name is called as plasmid pBR322. This was one of the first artificial cloning vectors to be constructed and undoubtedly the most widely used cloning vector created by Bolivar and Rodriguez. It was created in the 1977 in the laboratory of the Herbert Boyer at a university of the California, San Francisco. Already we know very well about this concept, isn't it? These things also. The P is stand for plasmid and BR is for the Bolivar and also Rodriguez. Here the P is denoted as the plasmid, the vector name, extra chromosomal body. And then BR is the scientist name also included in the plasmids. So researchers who construct it. Okay. So these are the things already yesterday we saw it. Today, so remaining things, see that one, the BR322, the B is denoted as plasmids. So here you have two things. And BR, Bolivar and Rodriguez. The 322 is nothing but what is the number of plasmids, otherwise called as distinguishers here. What are the differences between? How it will be differentiate or distinguish when you are giving the number 322, 323, 325, 27, 7 like that. There are some differences, distinguishers is there within the molecules. That's why they are giving the different numbers. BPR322, BPR325, BPR327 is also present. Okay, according to their differentiation, they may be classified, they may be given the name. Okay, so in this plasmids, how many antibiotic resistant genes are present? Already we discussed. One is called as two types of antibiotic resistant gene also possesses. One is called as ampicillin resistant, is simply called as AMPR. And the other is called as tetracycline resistance, simply called as TET or genes. These are the two genes is present. Not only that, and also some of the enzymes also, encoded genes also present in it. See that one. So these are the things. See here, this is the exact structure of the BPR322. Okay, see that AMPR. So this portion is considered as a AMPR. But in this category, PST1, PUV1, and also EcoRI1, CLA1, HindD3, and BAMH1, SAL1, okay, and PVU2. So like that, they mention different genes also. The AMP resistant, which one is responsible? These two is responsible. Tetracyclines, you can take an example, so BAMH1 and also SAL1, Salmonella. This is also some of them now. Okay, like that, they marked. They were marked, isn't it? See that one. So it's the same thing is here also. The next one is, see that one. So this is also the name of the scientist already in the year 1977. BPR322, the B denoted as plasmids, BR denoted as Bolivar and Rotricus, 322 number of distinct users. Suppose here 325 is there, so it is also different between the 322 and 325. So what about these two? 325 and 327, etc. have been identified and reported. Many of the plasmids have been reported in BPR325 only. Suppose 322 is not used, instead of 322, they were used 325. Don't forget, this is also one of the questions already asked in the NEET. Okay, my dear. Okay, see that one. So ampicillin resistance gene, how it will be? The ampicillin resistance is denoted as AMP resistant R. Okay, AMPR. This gene does code for an enzyme that modifies ampicillin into a form that is non-toxic to Escherichia coli, simply called as E. coli. And the tetracycline resistant gene is a set of gene coding for enzyme that detoxify the tetracycline. So these are the very, very important. So where there is a gene is present, the gene is resistant to the ampicillin, means they produce some enzymes resistant to the ampicillin. Likewise, the tetracycline resistant gene also produce some enzyme that is also detoxify the tetracycline. So these are the two enzymes also very, very important. The next one is how it will be constructed, how many genes is there, what are the things present in it. The BPR322 gene is, see that 4,361 base pair in length. Okay, this 4361 base pair in length is considered as a ROP gene, encodes ROP, repressor of primer protein. Okay, repressor of primer proteins. The repressor or primer protein genes code for the proteins involved in the replication of the plasmid. That's why it is called as ROP. Okay, repressor of primer. Okay, what is the main functions of the ROP gene? The ROP gene, what is happening? Replication of the. So protein involved in the replication of the plasmids. It is very, very essential one. Once again, repeat for you. That PPR322 is, it has a two resistant gene is there. One is called as AMPR, another one is called a TTR. Okay, corresponding enzyme also secreted and detoxification takes place, non-toxic and detoxify. At the same time, the BPR322 gene is, what is that? How many base pair present in it? 4,361. Okay, and length. So that gene is called as, what is it? ROP gene. That ROP is denoted as which one? Repressor of primer. R, O, and P. Repressor of primer proteins. Not only that, ROP genes code for the proteins involved in the replication of the plasmid also. That is very, very important, my dear. See that one. The following is the list of some restriction enzyme and their sources. So already we discussed not only that resistant gene and also some of the other restriction enzymes also present or encoded by the gene present in the BPR322. What are they know? See that PVU1, that is denoted as a Proteus vulgaris. So simply called as PBU. P, see the first letter, vulgaris, VU, that is number denoted as 1. Okay, and then PST, Providencia as 2 or T. So this is another one. Providencia, 2 or T, another one. And Sol 1, Streptomycin albus, S denoted as S. See that the first letter is denoted as the genus name and species, like that. Just imagine the first letter of the P here, the ST is the genus number. Okay, the letter of the first 2 letters from the genus. Here Sol, S denoted Streptomycin albus. So this is the first AL. SMA1, Ceritaria and Marcescens. Ceritaria, Marcescens 1. All are having 1, 1, 1, 1, isn't it? Cla 1. Crypanon latum. Cryopanon latum. So like that, these are the enzymes also present in it. These are the enzymes producing genes also present within the pBR322. So just note it down, my dear student. 2P, 2S, 1C. Okay. See then VU vulgaris, Proteus vulgaris, PSD, Providencia statui, and Sol 1, the Streptomycin albus, and SMA 1, Ceritaria and Marcescens, and Cla 1, that is called as Cryophenon and Latum. So these are the enzyme producing genes also present in it. There are improved derivatives of the PP322 and example BP325. So from this one, 322 is a precursor for the development of the improved varieties, the derivatives for the 325. So this is considered as a mother. Okay. From this one, they may alter the new one. So the number will be also changed. Here 322, here 325. So it is a improved plasmids. As an additional a chromopenicol resistant gene. Here, what is the difference between these two genes, these two plasmids, 322 and 325? Like that, they may ask the question, which one is the addition? Chromopenicol resistant gene is also present in the 325 number. 325, the plasmids, compared to the 322. Containing a unique EuCO-RI one side. This is very, very important. EuCO-RI site. So, okay. So this is the important points. See that one here. EuCO-RI. So in this part, okay. In this part, what happened? There is a chromopenicol in this part. See that one. So in this part, there is a one additional is the name, the number is also changed, 325. See that 325. Okay, I didn't pick that one. So next classes show you. And the next one is small p, capital U, capital C vectors. PU, say plasmid UC vector. This is denoted as a plasmid UC vectors. The name plasmid UC vector is derived because it was developed in the University of California. Okay, see that one. The University of California is denoted as UC by Messings and his colleagues. So this is the name of the scientist in the year 1983. When it was discovered, that the BPR322, 1977, 1977, this one is 1983. What is the difference between these two? How many years? Dhanalisimi, 1977, 1983. So in between, how many years is that?
6 years, sir.
6 years. Very good. So you can easily identify. Then only it can be easily remind at the time of examination point of view. Okay. So 1983. Okay. So this is very, very important. Who discovered this one? PUC plasmid. What is that? University of California vectors. Discovered in the year 1983. Who discovered? Messings and his colleagues, co-workers. Okay. So go to the next one. These vectors containing AMP resistant, that is ampicillin resistant gene, and a new gene is called as LAC-JET. Which one you know? The PUC. Don't forget me. Here the addition one is the LAC-JET. But here is absence of which one you know? Tetra. Instead of tetra, which one is present? LAC-JET is present, which was derived from the lac-opron of E. coli. Already we studied Jacob and Mona discovered the lac-opron concept. Okay, from the E. coli, that code for an enzyme. That is very, very important, my dear. Okay, so here the addition gene is also present. That gene is called as LAC-JET. Okay, so it is also derived from the lac-opron of E. coli. So the E. coli bacteria, it has an extra chromosomal body, and the particular gene is responsible for LAC. That gene was isolated from the E. coli and also inserted in the, which one you know? The PUC. Okay, so 19 plasmids also. They have given in the name is also 19. See that one. The PUC 19 plasmids in an E. coli, the plasmids of 2686 base pairs that contain the replication origin and ampicillin resistant genes and a LAC-JET gene for beta galactosidae. Already in our, but already we studied there are two structural genes there. Which one you know? JET and Y, and also another one is called as A. JET is responsible for the beta galactosidase, and Y is responsible for the, which one you know? Permeases, and the last one is called as what is the transacetylases. So the JET gene is responsible for beta galactose. This was isolated from the E. coli bacteria and also inserted into the PUC19 plasmids. It has this molecular structures. This is very, very important. Okay. 11 restriction sites also cluster within the LAC-JET gene. How many restriction sites is there? 11 restriction sites is there. Don't forget, my dear student. This is very, very important in this plasmids. Okay, compared to the BP322. Okay, there are two plasmids now we were studied. One is ampicillin resistant, another one is tetracycline resistance. How many nucleotide is there? Who discovered when? But in this category, and also that plasmid, which one you know? University of California, the name is called as 19. Why they are giving 19? Because it's a 19th one. So 20, 21, 22, 20, like so many things that were discovered. Particularly, they mentioned this one is. This is also one of the plasmids isolated from the E. coli. It has a specific base pair 2686. Base pair containing replication origin. Only here what happened? No, there is one of the additional gene is called as LAC-JET gene. It is also isolated from the lac-opron concept. That is called as beta galactosidase because the JET gene is responsible for the production of the beta galactosidase. Okay. In this plasmids, how many restriction sites also present in it? 11 restriction sites present in it. Don't forget. These are the 4 or 5 points is important for the NEET point of view. For the next one, TI plasmids. It is another one already we studied in the what are the types of plasmids already we discussed general outline only. Okay. Now we will study the TI plasmids. Already told you. I already told the answers also. Tumor inducing plasmids, simply called as TI plasmids, is nothing but the tumor inducing plasmids. This plasmid is responsible for which microorganism, Agrobacterium tumefaciens. Don't forget, TI plasmids is also, what is that? Agrobacterium tumefaciens has been treated as a nature most effective plant genetic engineer. Okay. So the large plasmid in this agrobacteria are called tumor-inducing plasmid. That's why it is simply called as TI plasmids. And also another one is also, this one is called as tumor-inducing. Sometimes they may be used for the root-inducing plasmid. That's why it is called as RI plasmids respectively. Once again, repeat for you, not only the TI plasmid and also RI plasmids is also there. The TI plasmid is nothing but the tumor-inducing plasmid. RI plasmid is nothing but root-inducing plasmids. Okay, see that one. Which confer the pathogenic capacity on their coast, the Agrobacterium tumefaciens bacterium invade plant tissues, causing a cancerous growth called ground call effect. Here you must note it down. The ground call effect was induced by the cosmetic organisms. They may ask the question that is called as Agrobacter tumefaciens. One. Second one is the ground call diseases, otherwise called as cancer disease, caused by the which plasmids, okay, produced by the Agrobacterium tumefaciens, TI plasmids, and also RI plasmids also there. Okay, these are the two things. Sometimes the cancerous cells produce in the upper part, that is called as above the soil is called as shoot system, and the lower part is called as root system. That's why they have given us a different, okay, TI plasmid and RI plasmids. See that? So you will get the effect ground call disease due to the ground call disease is caused by the microorganism Agrobacterium tumefaciens, which plasmid is responsible TI plasmids, tumor inducing. Tumor is nothing but the cancer. Okay, media. See that one. So this is the plant. Okay. So there is a soil already in our above the soil. What happened? There is a root system is the shoot system is there, and below the soil there is a root system is there. Automatically see that this is the considered as a tumefacian microbacterium. Okay, agrobacterium, isn't it? This bacterium, what happened? It induced the ground call effect. Ground call just like a cancer cells, just like a tumor, just like a mass of cells. Cell division undergoes. As a result, what happens? You will get the tumor cells. Finally, what happened? It may block the passage of the any other things. Suppose if it is disease, means what happen? The product and whatever it may be do not get easily. You will not get the good result. That is the reason. See, this is the plant, the ground call effect. Okay. Some of the plant, this is the croton plant, I think. Okay. It has some effects. So many plants it contain. This is the effect due to the tumefaciens. This bacterium infect all both leaves, agricultural crops such as the tomato, soybeans, sunflower, and cotton, but not cereals. Don't forget, my dear. This is also very, very important. This is exceptional, isn't it? Cereal not suffer by the Agrobacterium tumefaciens, otherwise called as TI plasmids. Other than which one? No, cereals. What are the plants? They may be affected by this. No, the tomato, soybeans, sunflower, and cotton. Okay, this is very, very important, my dear. And the next one is TI plasmid is a large circular DNA, the molecule up to 200 kilobase pair in length. This is also another important TI plasmids. Okay, the kilobase pair should be 200 kp in length. Okay, then next, what will be the mechanism? See that how it will be act on the plant. During infection, a part of the TI plasmids called tDNA is integrated into the plant chromosomal DNA. Already we know all about it. That things also, isn't it, my dear? Okay, taken examples of any one of the, what is that? Bacteria. So this is considered any one of the bacteria. Inside, about there is a plasmid is there. The particular area, this is the plasmid, isn't it? This is called as, what is that? This is plasmid, isn't it? Plasmid, isn't it? That plasmid is also isolated in the outside. Okay, this is considered plasmids. Particularly in this part, we can mark it in different, then only it will be better for you. And in this part, TI plasmids. Okay, TI plasmids. So what is the meaning of TI? That is denoted as tumor inducing. Tumor. Okay. Tumor, MOUR. Okay, tumor inducing plasmids. Inducing plasmids. Okay, tumor inducing plasmids. This can be what happened, when it incorporate with the plant DNA. This is considered as a plant. This is plant DNA. Okay, this is considered as a plant cell and also having the molecule, this one. Okay. This TA plasmid, this portion, this portion. Okay. This portion is when it incorporate with this plant DNA, as a result, what happened? The plant containing the ground call effect, the plant, when these two genes are united together in the plant cell DNA molecules, as a result, what happened? You will get the ground call effect. So imagine this is considered a leaf. Okay. So this is the leaf. Okay. This is considered a leaf, and then effect will be very complicated, isn't it? So maximum in this portion. Okay. The cells are produced like this, and also some of them are having like this site. This is called as, what is that? Ground call. GROWN, ground call effect. Okay. What is the responsible for ground call effect? Mean TI plasmids. When the TI plasmids mean tumor inducing plasmids of any one of the microorganisms, particularly which bacteria? Agrobacterium tumefaciens. That bacteria, the TI plasmids, when it incorporated with the plant DNA molecules, as a result, what happened? The ground call effect is induced. Okay. So this is very, very important idea. This is a very, very important things. Okay. See that one. Okay. Yes. Okay. So this is very, very important. So next, the tDNA containing genes coding for the protein that stimulate the divisions of the infected cell, and also coding for enzyme that convert the amino acid arginine into unusual derivative, generally no plane or octopine, that the bacterium need in order to grow. tDNA has 13 genes. Here is noted down what is by T gene. The T gene contains genes coding for the proteins. Okay, that stimulates the divisions of infected cell. Which cell is infected by the, which one? TI. Okay, T gene. What happened? It automatically, when the foreign particle enter into the body, what happened? Automatically the body produce their protein molecules. Already know very well about that. Is called as, what is that? Antigen. Okay. So infectors and also coding the enzyme that convert the amino acid, particular which amino acids also converted, the arginine converted into nopalene or octopine. So this is the conversion. Arginine converted into nopalene or arginine converted into octopine, that the bacterium need in order to grow. So then only what happened? It will be survive. Okay, this is very, very important. When it enter into the cell, plant cell, the amino acids also present inside. The amino acid, which amino acid? Arginine is the amino acid. The arginine amino acid is converted into unusually derivative, generally, what is that? Converted into nopalene and also octopine. This conversion is very, very important for their growth. Okay, which growth? The bacterium growth. Which bacterium? Agrobacterium tumefaciens. Okay, so that one. The tDNA is contained how many genes also present in here? The 13 genes also present in there. The PUC is 11. Okay, UC, isn't it? University of California gene contain, the plasmids contain 11 genes. But here 13 genes is there. Don't forget, my dear. This is very, very important. See this one. Agrobacterium mediated transformation. This is the bacteria, already in our board. The bacteria inner side, there is a invisible DNA molecule present in it. We cannot be able to visible. So it is called as, what is it? Prokaryote, isn't it? So addition with it, there is a extra chromosomal body is present in it. This extra chromosomal body is called as a plasmid. That plasmid is called as T plasmids. When the plasmid is isolated outside, we can rupture the cell with the help of some mechanical equipments. We can isolate the plasmids, particularly this portion is responsible for the TI plasmid, tumor-inducing plasmid. This plasmid is called as what is a tumor-inducing plasmid. The tumor-inducing plasmid, we may cut the restriction enzyme with the help of the restriction enzyme in this part. In this part, we are cutting here. As a result, we will get this like this fragment from this portion. Plasmid is cut using the restriction enzyme. The restriction enzyme only cut made in the which area? Polyandromic sites only, this portion. Okay, cut and removed it. After that, what happened? The foreign DNA cut with the same restriction enzyme and insert into the plasmids. So after the we made a cut, isolate the particular desirable gene, then it may be inserted into the host DNA. So which one is the? This one is A, another one is B. Okay, this is the A gene. Okay, imagine this is the vector gene, isn't it? It is also inserted into the another recon. So when you are adding the A gene as well as this one is also taken from the another genes. This is the animal gene, isn't it? So this one is obtained from the plant cells, this genes, this portion. So both are united. That condition is called as recombinant plasmids with the help of the ligase enzymes. After the recombination plasmids, again what happened? No, this setup, complete setup, what happened? Inserted in the same one. So this one, bacteria, that is called as E. coli. After the recombination completed, what happened? It can be reinserted into the bacterium, reinserted with the help of any one of the gene gun, okay, micro pipette, okay, biolistics, whatever it may be, any one of the method they were used for the laboratory aspects. After that, what happened? It inserted means both the gene, these genes present inside. That means A gene as well as the B gene present inside. So automatically, that multiplication takes place inside it. tDNA carrying inserted gene incorporated into the plant cell chromosomes. There is a reason. Okay, and also inside. Then the plant cell culture. Suppose if it is inserted into the plant cell, what happens? Automatically the plant cell undergo the culture process with the help of the tissue culture. After the tissue culture, what happened? Both the gene properties present in tissues. So it's also after the growth, what happened? The transgenic plant expressing inserted gene is generated. So this plant is, what is that? A transgenic plant. Okay. Likewise, there are so many plants nowadays they were discussed by China. Already you know very about. Any one of the pumpkins, take an example, maximum weight will be 5 to 10 kg only. For example, I'm telling you. Suppose in Chinese people, they were discussed a transgenic plant means that weight will be 25 kg to 50 kg. Even sugar cane, even rice, even take an example of potato, tomato, whatever it may be, mango is also there. These are all considered that it is transgenic plant. It's also one of the biotechnology method or genetic engineering methodology. This is the way to produce the new plant. So these are the important things. Are you understand, Dhanalakshmi, now? Do you understand?
Yes, sir. Understand.
Yes. Yes. Are you follow it now?
Yes, sir.
Okay. So what about the next one? So the tumor-inducing plasmids of Agrobacterium tumefaciens is modified into the cloning vector, which is no more pathogenic to the plant. Tumor-forming genes have been deleted. So as a result, what happened? The particular tumor-inducing plasmid will be removed means what happened automatically? The tumor-inducing gene is not present inside. So the tumor-forming genes have been completely deleted in the generation. They used to deliver the genes of our interest into a variety of plants. This is also very, very important. The tDNA with desired DNA spliced into, it's inserted into the chromosome of the host plant cell, where it is produced copies of itself by migrating from one chromosomal position to another at random. This is also very, very important. After this one, we can use the another chromosomes. It can be randomly what happened. It can migrate from the within the cell. Okay. This is the important things. The transferred into the soil, the plant lets grow onto the mature plant. This is also already we did in the tissue culture technology. Already you know about how it will be tissue culture is possible. Can you understand that one? How will you tissue culture? How will you do? Okay. So how will you do the tissue culture media? Okay. Tissue culture means you know about. First of all, you should know the plant cell is there. Already you know about. Taken examples. Again, same process is there. There is a plant. Okay. The plant is also made up of more than number of cells is also there. Already you know about. Okay. This is very, very important, isn't it? So the plant cell. So you can take an example of this is considered as a plant cell. Okay. The plant cell, it has a totipotential characteristic. What is meant by totipotential means? What is the meaning of totipotential, my dear? What is the meaning of totipotential? Any idea? Dhanalisimi, do you know what is my totipotency? Take an example. This is another cell. Also, this one is also another cell. Take an example. This is also cell structure. This is the nucleus. Okay. This is a plant cell. Okay. Okay. So this is a cell. Normally, you know about. From this cell to how it will be used, the tissue culture technique. What is by tissue culture? Will you please? Tissue culture. What is by tissue culture? How it will be possible, you know about that? You have to take the one of the cells. This is it has a ability to produce the entire plant. The single cell, it can produce the entire plant is called as totipotency. That ability is called as what is a totipotency. Okay, this is called as potency. Okay, totipotency. How it will be done, you know? So one of you know about the first of all, you should take the tissue culture methodology. You should know the any one of the what is it test tubes. Okay, take a test tubes. Not only that, this is considered as a test tube already, you know, but inside the test tubes, what happened? There is a culture medium is there. That culture, that specific, what is that? That is culture medium. Already, you know, this portion completely filled with the medium. So, agar medium, already, you know, report. Okay. This portion is filled with the nutritive substances, simply called as, they are all collectively called as, what is that? Agar. Agar is also isolated from the one of the brown algae. Agar medium. Agar, isn't it? Agar is a nutritive substance medium. Okay, so agar medium. Okay, so this is very, very important, my dear. Agar medium is very, very important for the growth of the any plant tissues inside the cells. So here what we have to do, you know, this is the and also having the nutritive substances. Agar medium, it contains the sugar and phytohormones, minerals and nutrients, so on so there, and specific pH is also there. Very, very important things also. Don't forget, my dear. After that, what happened now? We have to take the plant, let's say, from the any one of the plant, what we are going to culture within the cells. We have to place it in aseptically. Okay. So inside what happened? Inside what happened after that? They are all are we have to do in the aseptic manner. Aseptically. This is very, very important. That is also, it's also covered with the, there is a, which one, you know? There is a cotton. This is considered this is a cotton, completely closed the things. Okay. And also kept under the, what is that? A specific place. Okay. So they need light. So they are very, very important. So when you are giving the light, what happen automatically inside there is a cell is there, no? The cell is also undergoes the multiplication by the absorption of the nutrition from the agar medium. Here you take the nutrition from the agar medium. So as a result, what happened? The test tube inside what happened? There are numerous cell formation takes place. This is considered as a numerous of cells. Okay, this mass of cell is called as what is that? This mass of cell is called as callus. CALL. What is that? Callus. Okay, just imagine this callus is undifferentiated, all are unique in size. Okay, and after that also what happened? It's also transferred. Okay, transferred to the another things. When you are adding the another phytohormones, when you are adding the phytohormone, what happened? Within the test tubes, what happened? Just imagine. So within the test tubes, what happened? Just imagine. So within the test tubes, okay, within the test tube, how it will be? Just imagine. Okay, the same process there. Okay, within the test tubes, how it will be? Just imagine. This is covered with the which one? No, cotton. This one is also covered with the cotton completely. So just like this. Okay, this is completely covered. Inside already you know a little bit there is a nutritive substance is there. Already we know a little bit this is the what's the plant. Let's see, this is the callus, isn't it? The callus is also grow. It produces roots as well as a shoot system. This is the first plant. Let's see how it will be. Just imagine like this. Okay, the mass of cell, it produces the first, what is that? Like this. After that, not only that, here number of cells, we cannot be able to calculate numbers, number of cells, thousands and thousands of cells there. We can isolate all those things. Okay, we can isolate, and also again it's also inoculated in the aseptic condition in different what is that? Test tubes. Likewise, each and every rack, thousands and millions of test tubes we can place it. So finally, what happened? This is within the test tubes only. After that, it's also undergoes the process called as hardening process. What is the meaning of hardening? Means inside the plant, let's see, once the root system is also originated, after that is also planted outside the which one? Land. That condition is called as what is it? That condition is called as what is that? Hardening. That process is called as what is that? Hardening. So take an example. So this is considered as a land. Okay, after the complete of the root as well as the shoot system formation takes place, this can be replaced in the where in the land. Okay, it can be transferred to the land process. Okay, so this process is called as what is it? Okay, just imagine this considered as a what is that? Hardening process. Hardening from the test tube to hardening. Okay, this is considered as a hardening process. When so after the tissue culture, okay, there is a small plant, let's say any one of the plant tissues, whether flower, seeds, whatever it may be, whether it may be Anthridium, Orgigonium, whatever it may be, any one of the body can be closed, inserted into the test tubes. Okay, it grows as a mass of tissues that is called as callus tissues. So after the formation of callus tissues, also undergo the further inoculation process. So many inoculation process also. Then only you will get the pure plants. Suppose we did any mistake in this part, the next it will be corrected. Likewise, after the 10 test tubes, you have to take the 10 test. This is the first one. Consider as the first one means you have to number up to the 10 test tubes in the line. Here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, this is considered as the 10th test tubes. Okay, 10th one. This is sorry, this is not a 10th one. Okay, so this one is 10th. So the callus is there. After the callus, it's also undergo the. This is the first test, and also here you take the one to another one. So likewise, you have to transfer from 1 to 2, 2 to 2, 2, 3 to 4 to 5, 6, 7, 8, like that. You have to what is that? Change the callus into the another medium. So 10th one, you will get the pure, what is that? Plantless, pure form of plantless. After that, it just leave it as it for the few days. What happened? No, the root system as well as the shoot system formation takes place. Root system is called as the origination of root as well as the shoot within the test tubes. Once this step is completed, it may be transferred to the which process? Hardening process. In the hardening is nothing but the plantlets are grow into where transfer into the land, from the lab to land. This process is called as hardening. That things has given here likewise see that here also they told the same process. After that, the inoculation process completed, that can be transferred to the, what is that? Outside the plant. That is called as, what is that? See that? This one is considered as a knee plants. The carrying for the foreign gene, they express throughout the knee plant. See the transfer into the soil, the plantlets grow into a mature plant. Okay, the plantlets means a young one, isn't it? The test tubes, it has a young plant, otherwise called as plantlets. It can be transferred to the soil. Soil means what is that? That process is called as hardening, I told you. So after that, what happened? The plantlets grow into a mature plants. They're carrying the foreign genes and expressed throughout the new plant. That's why that a mature plant, it contains both the genes. That is one, that is the reason. So it fight against many diseases, whether it may be resistance of the tetracycline or ampicillin or whatever it may be, any other things is there. So likewise, similarly, the retrovirus have also been disarmed and are now used to deliver a desirable gene into the animal cell also. Sometimes take an example. Okay, but it no longer produces tumors. Such plant cells are then cultured, infused to the multiply and differentiated to form a plantlets. It is a short period. No longer produces tumors. Longer produces tumors, it may be very shortened period. It produces tumors, such type of plants and animals also cultured. Okay. Transfer into soil, that plantlets grow into mature plants carrying the foreign gene and expressed throughout the new plant. Similarly, retrovirus have also been disarmed and also now used to deliver the desirable gene into the animal cell also. Okay, plant to animal, animal to plant, like that. See that? Plant and animal viruses. How it will be? What are the plant viruses? What are the animal viruses? So virus also, otherwise called as bacteriophage, is also act as a vectors. That's why it is called as viral vector. Okay. See that one? In recombinant molecular biology, the recombinant means both the genes, different genes united together in the molecular level. Okay. The virus particles have been modified to be used as a carrier of nucleic acid into a cell termed as a viral vector. Sometimes they were used as a viral, okay, virus. Bacteriophage also act as a vectors. It carry the molecules of the any one of the nucleic acid. See that the viral vector are highly efficient in transferring target DNA, RNA segment to the host cell with high specificity. This is very, very important. And wild type of viruses are modified by deleting the non-essential genes and incorporating exogenous nucleic acid segment to the construct a viral vector. Same as it is the E. coli bacteria. What we did, the same process also we have to do in the where? The bacteriophage, addition, deletions, selection one, and also remove it. Which one is the desired quality? You have to insert it. But the cost is which one, the bacteriophage is a cost. There what we have to take, in the previous experiment, we took which one, E. coli as a host. Here, they were used which one, virus. That is the reason. So, the virus inside what happened, automatically, incorporated with the viral genes. So, it can multiply within the viral. So, that's why it is considered as a viral vector. So, the viral vectors have a wide application in gene therapy. This is very, very important. And the targeted drug delivery system, where it is mostly used, drugs delivery system, mainly advantage of the viral vector or high transfer efficiency and high cell specificity. Sometimes they may ask the question, which one of the following is a highly efficiency and high cell specificity? Like that, they may ask the question true or false, match the following. And sometimes they may ask the question, direct question. Sometimes it may assessment and reasoning. Okay. Likewise, they were asked the question, you should identify this part is very, very viral. Vector is have a wide applications in gene therapy. This is also very, very important. Gene therapy. What is the meaning of gene therapy? Desired quality will be, desired gene will be, we can induce inside the virus. Okay. The multiplication already in our body, how it will be. See that the commonly used viral vectors and their properties. Here there are, this is present in your book. The viral vector, what are the viruses that? This is called as adenovirus, retrovirus, lentivirus, adeno-associated virus, the herpes simplex virus, simply called as HSV. Here, see that adenovirus, this is an animal virus. Note it down, my dear. DNA. Retrovirus, it is also animal, but nucleic acid is different, RNA containing. Lentivirus, RNA. Adeno-associated virus, DNA. So herpes simple viruses, this is also animal virus, DNA. So here the 3 DNA molecules have been given and also 2 RNA molecules is also examples. Each one had a specific insert. See the 2 to 8 kp, 2 to 8 kps retrovirus, 7 to 18 kps, 4.5 kps, and less than 30 kps. These are the, what is the inserted things. See the next one, you can see the same things is also given here. See the inserted size. Okay, adenovirus, the DNA molecule, and 2 to 8 kp only the inserted size. Retrovirus, RNA is also 2 to 8 kp. Lentivirus, RNA, 7 to 18 kp. Adeno-associated virus, this is also, it has a DNA virus and 4.5 kp. Okay, and herpes simplex virus, HSV, DNA, and also less than 30 kp. So these are the important ideas. This is very, very important. Just note it down. So this is considered as a retrovirus. Retrovirus means what is that? HIV is also one of the retrovirus. Already know about. Inside what happened? There is a, suppose if it is a RNA molecules. Okay, suppose if it is a RNA, retrovirus is the RNA molecule, isn't it? So RNA molecules present inside. So HIV is also one of the retrovirus. It contain which one, envelope. The outermost covering is called as envelope. Okay. The envelope is also made up of glycoproteins. There are 2 types of glycoprotein have been reported. One is the 121 and glycoprotein 121 and glycoprotein 42 or 40. So that one is also present in it. And inside there is a one of the covering is also present. Inside there is a one another covering is also okay. And then the inner side also there is a 3 layers. This is the outermost layer. This is the middle layer. This is the inner layer. Inside the layer, what happened? No, there is a RNA molecule present in it. RNA means single standard RNA molecule present in it with 2 copies of reverse transcriptase enzymes, because it's an RNA molecule, isn't it? Nucleic acid is here, RNA molecules. So this is capsid. This is nothing but the protein molecule. See that this is the capsid. Capsid is nothing but the protein molecule. Virus is composed of the protein as well as the nucleic acid, both only present in it. These 2 only present in it, the protein molecule as well as nucleic acid, whether it may be DNA or RNA molecule. Okay, the outermost covering is also, what is that? Protein molecule, simply called as capsid. And this capsid is also made up of capsomeres. When you are studying the TMV, tobacco mosaic virus, you can see it. Okay, so the reverse transcriptase enzymes present in this is the structure of retrovirus. See the next one is, so this is also a retrovirus, isn't it? It's a type of retrovirus. And then see that herpes simplex HS virus. See that one, the lipid envelope present outside. There is a lipid virus, a lipid envelope. It is also made up of fat. Okay, so envelope is there. Not only that, there is a hook-like structure, different types of hook-like structure. These are all are considered as a envelope of protein molecule, that is glycoprotein GN. GBGN molecules. tegment. Tegments is the in between the molecules, the in between the inner portion as well as the outer portion. There is a middle portion is that middle portion is called as tegment. Nucleic acid. Capsid is nothing but the protein molecule. The nucleic acid is the nucleic acid surrounded by the protein molecule. That is called as what is a capsid. Inside there is a DNA molecule. The DNA molecules already in our level. It's nothing but the double standards molecules. Helical structure is there. The structure is isolated and given the structure like this. This is the entire exact structure of the DNA molecule. Here the regulation, capsid assembly, envelope protein, DNA replication, all those things present in linearly arranged. Already you know all the genes are arranged in the linear manner. Already you studied that things also, isn't it? This is very, very important, my dear students. And the next one is called as adenovirus. Okay, this is a adenovirus. Structure will be see that one. Same, similar to that of the retrovirus, but the hooks is also present in it. Okay. And then this is also one of the adenoviruses. It is also same, but these are different structures. Okay. And the next one is called as the lentivirus systems. This is considered similar to that of the HIV virus. But here see that there is a outer protein molecule, the surface, glycoprotein 120. Okay. See that transmembrane, glycoprotein 41. So this protein is called as 41. This protein is called as what is then, glycoprotein 120. I already told you 41 and 120. The same structure is also present in the HIV also. Okay. This portion is called as MA, otherwise called as matrix, and PR means protease. This is nothing but the enzymes. This is enzyme. Inside there is a enzyme is there. And also this is called as integrase is also one of the enzyme. Integrase means unity. When you are combined with the other molecules, it can be easily integrated. Integrated means it incorporated with the another nuclei. See the SS RNA. SS means single stranded RNA molecule present inside. This the green color structure is single stranded RNA. See this that orange color. This is see that orange color indicating the reverse transcriptase enzyme. Okay, NC nuclear envelope, CA capsid, VIF, VPR, NF. These are the some extra particles. This is the entire structure is called as what is the lentivirus system. Completely, it is nothing but a specific systematic aspect. Okay, so this is when it enter into the any one of the foreign particles. Okay, this is considered as a foreign particle. When it invade the microorganism, the animals or plants, what happen? Automatically, what happen? The RNA molecule produce the DNA molecule with the help of the reverse transcriptase enzyme. As a result, the overall control of the caused by the invading foreign pathogens. So this is very, very important. Adeno-associated virus. Okay, this is the adeno-associated virus. This is considered as a adeno-associated virus. The co-transformation, infection, and help virus, AD, helper viruses. Different types have been given. See that one? It's a very big sizes also. And we will finish it up to this level only, my dear. We will see the remaining things on the next class. Simon's virus 40 was the first virus used as a cloning vehicle in mouse cell. Simian. It is a DNA virus that can infect human as well as other mammalian cell lions also. We will finish it up to this level, my dear. So we will summarize all those things. Already we discussed what are the things, See that one. See the pBR322. Already we know about what are the content present in it. Amblycellule resistant gene, and also another one is called as tetracycline resistant gene is also present in it. Okay. And also the how many gene base pair present in 4361. That's in 4361 base pair length. And rope gene is there. Rope gene is called as repressor of primer. It's also present in it. It's used for the replication of the plasmid. And then following enzyme gene is also encoded. That is called as the PVU1 and PST1 and PSOL1, SMA1 and CLA1, 2P, 2S and C. Okay, corresponding name has given here. The first letter of the genus name and second two letters of the species name has been taken as a short form. Okay, and then not only that, and the 322 and the 325 is also there. It is the 325 is an additional one is that there is chromopenicol resistant gene is present in it. It is also isolated for the E. coli 1. And the PUC is nothing but already but it is nothing but the University of California, the plasmid University of California. This was discovered by the name of the scientist Messing and also his colleagues in the year 1983. This is another one. So here what happened, there is a additional of gene is that is called as LAC-Z. Okay. So what will be the gene base per 2686? Okay. Easily identifications. Here the Z gene is the gene is responsible for the lac-operon. Lac-operon means already in our Z gene responsible for the synthesis of the enzyme is called as beta-galactosidase. Don't forget, my dear. This is very, very important. Okay. And TI flam is already in our but agrobacterium tumefaciens, and also another one is called as root inducing plasmids. Also both are inducing the tumor. Okay. So as a result, what happened, the ground call effect is there. Ground call effect also done by the induced by the which one, agrobacterium tumefaciens and the TI plasmid present in it. Okay. So that things already the structures and how it will be produced, all those things. Okay. And then, and particularly all the tomato and soybean, sunflower, and also cotton, but not in cereals. Cereals, it do not affect. At the same time, how many lengths it should be there, so 200 kp in length. Okay. This is also very, very important, my dear students. And then how many genes also present? 13 genes of the tDNA is also present in it. And also one more is that the amino acids also converted. How the amino acid of arginine converted into nopalline and also octopine. That is the reason for the tDNA. Okay. This is the entire process, how it will be entered, how it will be cut. We made a cut restriction inside nucleus. After that, what happened? It will be introduced into the course, and then how it will be, and also culture, and also it may be transferred to the hardening process. Okay. So already we saw it, these things is also. And then see that one, a plant and animal viruses. Now only we saw it, that things. A deletion is also there. What are the commonly used viral vectors and their properties? Maximum, this one is the very, very important one. How many size is there? Inserted size should be 2 to 8, 2 to 8, 7 to 8. Maximum, how much is there now? So that 30, below 30 only. So here, that 7 to 18 is the highest one. Okay. So that thing's also retrovirus, and also already we saw it, adenovirus is also, and also the lentivirus also system is also, adeno-associated virus also there, and simian virus 40 is also already we discussed. Okay, my dear. Thank you so much for the participation. We will see the remaining things on the next classes, my dear. Thank you. Take care. Bye. Good night.
Thank you,
ma'am. Thank you, ma'am.
Thank you, sir. Thank you.
Thank
sir.
you.